So good morning to all participants, and I would like first to thank the organizers of Biospain 2021 for giving me the opportunity to share my passion about cell therapy, as I've been in the field for more than 25 years. I spent 15 years at the beginning of my career in research and development. I had a chance to develop solutions for tissue and organ transplantations, as well as medical devices, closed systems, and specialty cell culture media to support the development of stem cell therapy products. And over the last 10 years, joining Merck Life Science, I've been more in a customer-facing role to work and partner in the field with cell and gene therapy companies with the aim to help them develop their manufacturing strategy. So the topic for today is about cell therapy manufacturing, and I aim to revisit some of our lessons learned over the last couple of years to explore the trends and also our drivers for success. I would like first to remind our participants from North America, most probably not joining early this morning, but maybe getting access to the record later, that the life science business of Merck, headquartered in Darmstadt, Germany, operates as MilliporeSigma in your territory, US and Canada. So let me start first with a brief overview of our playground and the market dynamic, which has been really strong. We all know that the cell and gene therapy road has been quite bumpy over the last couple of decades, but over the last couple of years, we see a very positive trend. So when we look at the data which were published 1 year ago by the Alliance of Regenerative Medicine, looking on the right side at the number of regenerative medicine companies globally with more than 1,000 players, which is great. The split across geographies says that around 50% of those activities and players are based in North America, 25% in Europe, 25% rest of the world, and rest of the world with a strong contribution from some Asian countries like Japan, South Korea, Singapore, and to not forget about China, which is known as the second largest country in the world after the US when we talk about the development of CAR T-cell therapies in oncology. So there's a great number of people currently focusing on regenerative cell and gene therapy applications. And when we look on the left at the split across the applications, we see first that a lot of those activities still remain in early development phases. Phase 1 demonstrating the safety profile, phase 2 starting to look at the potency and potential clinical efficacy of those novel modalities. And this is true across the pipeline, across gene therapy products, meaning in vivo gene therapies, ex vivo gene therapies, meaning gene-modified cell therapies, also cell and stem cell therapies, and to some degree with a more limited number of applications around tissue engineering. But it's really interesting to see that the phase 3, so the late stage pipeline is quite robust these days. And with around 100 ongoing phase 3 clinical trials and regulatory decisions expected on multi-product candidates, we foresee a bright future in this industry. It's also interesting to remember that, and I keep that in mind, that FDA says that they are expecting around 10 to 20 new approvals per year in the near future, which is definitely a great news for the field. So all of that being said, it's interesting also to see that within the different categories of applications, cell and gene therapies, those are the cell-based immuno-oncology that makes up a growing portion of the pipeline. And I have listed on that slide a number of market-approved products which are CAR T therapies used to treat refractory cancers and targeting primarily hematological malignancies. That number of approved product is quite significant, and we all expect that from those marketed products, the field will demonstrate for the first time a successful business model and a profitable business model for the large pharma industry. Not surprising to see that the pipeline looks so promising that now we have large pharma players. We can name Novartis, we can name Gilead, BMS, and some others, really putting a lot of efforts to support these ongoing development programs to bring to the market those potential life-saving treatments and to develop what we name a potential path to cures, and especially in oncology. So strong clinical market dynamic, strong pipeline, and great opportunity for the business moving forward. I would like also to say a few words about our current pandemic context. Has been quite of a challenge over the last 2 years, as we all know. But it's also interesting to see that the pandemic has, to some degree, supercharged innovation. A good example we all know is the development of RNA-based COVID-19 vaccines that could demonstrate proof of concept of RNA platforms. And I take that opportunity to introduce our expertise and portfolio of specialty formulation lipids, which have been used to accelerate the development and manufacturing of these vaccines and novel modalities. Although in response to the ongoing global spread of COVID-19, several academic research centers as well as therapeutic developers have announced that they are investigating the application of stem cell therapies to treat this disease in the short term or to potentially address related complications in the future. One example is the use of mesenchymal stem cells. It is hypothesized that the cells could inhibit the cell-mediated inflammatory response associated with severe acute respiratory syndrome and therefore reduce the acute lung injury. So a lot going on in that space and quite promising. Those cell therapy programs have led to an increase in capacity requirements, which help the field move quickly towards more scalable, more sustainable production templates in bioreactors, in the example of allogeneic mesenchymal stem cell-based cell therapies, with the aim to reduce the labor and to improve the efficiency of the way we are producing the cell therapy products. For you to know that we do offer process innovation services to help you transition your process from lab scale to a stir-tongue bioreactor platform. So in case you're facing such challenges and needs for your programs, just feel free to challenge us and to contact me. So let's zoom on our topic for today and look at some complexity drivers and opportunities associated with them. So we see that therapeutic developers active in the regenerative medicine and advanced therapy space continue to advance their clinical program in a wide variety of indications with unmet medical needs. But as we saw, all of those programs, a lot of these programs, the vast majority of them still remain early development phases, where this is the time to make strategic decisions to build the manufacturing template for those products. So first of all, talking about complexity, we all know that we are dealing with advanced therapeutics, which are complex molecules, right? So innovative cell therapies require extensive manipulation of cellular materials like stem cells. If we take the example on the left side on that slide of the CAR T cell therapy products, the therapies educate the immune cells to target and to kill cancer cells. And to do so, it requires an integration into the genome of T cells of a new gene coding for a chimeric antigen receptor, what we call the CAR, which is also a complex molecular construct. It has an antigen recognition domain, extracellular. It has a transmembrane domain associated with an intracellular activating domain. So quite complex molecule to design this, what we call ex vivo gene therapy products or genetically modified T cell materials to treat cancers. The process of producing these CAR T cells has multiple processing steps, starting from the collection of apheresis materials, so the white blood cells, followed by the isolation of the T cell populations, then the activation of the T cells, followed by the transduction of those T cells using a viral vector to insert the CAR gene within the nucleus of the cells, followed by an expansion phase to multiply the number of CAR T cells. And at the end, there will be a downstream process to concentrate, wash, and formulate the final cell therapy product. So quite a lot of operations, quite difficult to manage, and associated what we see on the right-hand side of that slide with complex logistics as well. For autologous as well as for allogeneic cell therapy applications, the industry is currently focusing on a centralized manufacturing approach, and the centralized manufacturing approach requires to move materials back and forth from the site of collection to the site of manufacturing, the site for the treatment of these patients. And this had a lot of complexity around the logistics to move those materials. In addition to that, if we stick to the example of a CAR T product, there is a need to secure the supply of critical raw materials which are necessary to manufacture those cell therapies. And the two examples are definitely the production of GMP plasmids. Those GMP plasmids are used in the viral vector process in transient transfection to manufacture the lentiviral particles or retroviral particles, which will be used then as a critical raw material to engineer the T cells during the final manufacturing process. And in total, those are 3 separate manufacturing templates that you will need to develop to design a CAR T product if you decide to internalize all of these operations. Fortunately, there is a great opportunity in that space currently to limit the risk and your investments before any commercialization activities or any potential market adoption, which is the outsourcing of those manufacturing to experts in that space, meaning CDMO partners offering the requested expertise and GMP capabilities to produce those plasmids and to produce those viral vectors and potentially to produce as well your cell therapy product. And I take that opportunity to remind all of you that Merck Life Science offers world-class process development and manufacturing capabilities for viral vectors and virus-based therapeutic products from small scales, phase 1, all the way through the commercialization scale manufacturing and fill finish. Again, feel free to challenge us on those capabilities. Now back to the cell therapy bioprocessing, moving to the right side of that slide. We see that those processes require labor-intensive workflows, and those workflows result in high production costs, which is really quite of a challenge we all have to address as a community. If we look at this autologous CAR T process cost of goods component analysis on the slide, we can immediately see that the labor has a huge impact in manufacturing, but as well in other operations, including quality, media, preparations, also and logistics, but mainly in manufacturing, huge contribution to the manufacturing cost. So there is definitely an opportunity, a great opportunity to focus our efforts on developing manufacturing solutions that would simplify those processes and limit the labor associated with the manufacturing of cell therapy product, especially in a GMP environment. Indeed, I mean, today still, there's a lot of activities going on using manual processing steps, and which is quite understandable at small scale. If you have, for instance, to conduct some activities like doing a medium exchange in a T flask within a biosafety cabinet, it's quite manageable. It becomes quite of a challenge when transitioning to a scalable process in a GMP manufacturing environment. On top of it, let's talk about the time frame to make those process decisions. When developing cell therapies for life-threatening diseases with no real alternative treatments, there are expedited regulatory pathways to speed up your development and to speed up access to those novel modalities, which is great for the patients. It's a bit quite of a challenge on our side as manufacturers, because in that case, dealing with a compressed timeline for process decisions, even if it becomes a new paradigm, and which is great, compared to a standard clinical development program for biologics that I put on the top of that slides for with the example of the development of monoclonal antibodies, we see that those expedited regulatory pathways for cell and gene therapy products might reduce the time of development by 50%. Again, it's great for the patients, but those expedited regulatory pathways can lead to non-established templates, manufacturing templates that lead to a lot of inefficiency at the commercial scale. So this can explain why a lot of cell and gene therapy manufacturers do anticipate making post-approval changes in the process, despite the risk and cost associated with doing those changes post-marketing authorizations through running phase 4 clinical trials, when you have to demonstrate comparability not only in regard to the quality attributes of your product, but in regard also to the clinical efficacy. So the challenge for you and for your CDMO partner is to balance speed and quality throughout your shorter clinical development program and timeline, and ideally also balancing speed with a certain level of scalability and sustainability, as scalability and sustainability will become anyway the main drivers of your business when you are at the commercial scale. So we all know that at the very beginning, when starting a new development program, the drivers are speed, right? And this is driven by a couple of aspects or constraints or challenges I have highlighted on that slide. Speed to clinic, first to human remains the main drivers in early development phases, and both supersede our efforts to develop a robust manufacturing strategy from the very beginning. This can be due to our investors putting a lot of pressures as they are looking for meaningful clinical data generation with strong views on exit strategy. We might also argue that academic institutions, where most of those innovative cell therapies are developed, have limited process knowledge or possibly limited product knowledge from a quality and regulatory perspective to anticipate the need at the commercial scale. So our cell therapy bioprocessing group does offer, as I told you already, process innovation services in a PD environment to tackle those constraints and hurdles at early development stages and to support your organizations in the development of scalable manufacturing template early on. One example I can share with you, many times we had to postpone programs with stem cell therapies because they had first to concentrate their limited resources and budget and efforts on the generation of preclinical, preliminary and meaningful preclinical or clinical data to satisfy their investors, although they knew that demonstrating their ability to manufacture their cell therapy products at the commercial scale, or at least designing a robust scale-down model would be a major milestone to achieve and to value their development program and their business model. So with all of those complexity drivers in mind, let's now discuss how we can tackle them and leverage them to some degree as opportunities, and to look at designing the process from different angles. So we have seen that manufacturing of cell therapy products is complex. It's labor intensive and quite expensive. Also, balancing speed and quality and to some degree sustainability is crucial at early development stages, and defining the commercial end state of your product quite early in clinical development would support early process decisions towards more sustainable business model at the end. Of course, each project will have its own specificities and needs, if it's autologous versus allogeneic in regards to the scale, depending on the size of the products that you have to manufacture, the dose, the treatment, will be also depending on the size of your targeted patient population, etc. But there is one angle of the project, which is sometimes, I believe, underestimated, which is around building your product knowledge from early on. So product knowledge is not only key to understand your product, its quality, its mechanism of action, but it's also a great opportunity to really identify what I would say what the true critical product quality attributes are that you can use as a strong foundation to drive quality by design. I like to say a robust manufacturing process starts from the development of reliable product release testing method. I think this is true across all modalities in the pharma industry. And those analytical methods will become, as I said, a strong foundation to implement major changes in your manufacturing template moving forward when transitioning from clinical scale to commercial scale. So we all live in an industry where the process is the product, but I do recommend to not, how can I say, to not fall in the trap too early and to really claim that the process is the product only when your process supports the scalability and sustainability required to develop a successful business at the commercial scale, including for your analytics. Again, through our process development services, especially when designing processes in large-scale bioreactor, quite often we have been struggling with dealing with some variation around quality markers, transitioning from lab scale to large-scale manufacturing without any flexibility to move away from those little variations, although we understand we could demonstrate that those variations had most probably no impact at all on the functionality, nor on the potency of the cells, most probably not on the anticipated clinical efficacy. So my process is my product is great, but don't fall in the trap too early, and especially at early stages and small scale, keep in mind to keep some flexibility around your product knowledge and put some effort to understand what the true critical product quality attributes are and will be at the commercial scale. So another angle I wanted to highlight here on that slide to develop your manufacturing strategy is to design your process as simple as possible, or I like to say as complex as necessary, and even before thinking about closing, integration, and automation. Again, some lessons learned from our process development activities, as I already mentioned, doing a daily medium exchange manually, lab scale, it's very easy. Implementing such protocols at large scale in a GMP environment, in a bioreactor, in a closed manner, it's really challenging. So I'm convinced there is a lot to gain when challenging your process early on and your process workflow at small scale to identify all the steps which are not necessary, that can be removed or that can be combined to reduce the number of processing steps to a minimum, and so to reduce the complexity of future operations when you will be moving to a GMP environment. As I said, a robust manufacturing process starts from reliable product release testing methods and a strong testing strategy for cell therapy. I won't go through that slide, but you all know the 4 pillars around identity, purity, safety, and potency of the products, and there are many ways of approaching these biotesting methods. I just want to say that we recently did a survey and we asked a couple of cell and gene therapy companies to tell us more about the lesson learned from their early development programs and to understand what they would have done differently if we had to restart the development of those programs. And in many cases, those companies told us that they would definitely spend more time upfront in developing their analytical methods to build a much stronger product knowledge that would be used afterwards to consolidate their quality by design approach and to become even more stronger when developing the manufacturing strategy, especially at a commercial scale. So I believe the main challenge still resides around the potency of the cell therapy products, but again, be cautious with some quality markers which are locked too early at small scale and might become a roadblock at the commercial scales, especially for those that might be not relevant at the end. But you know, it's really important to keep that in mind, to be cautious around, again, this notion about my process is my product. Well, what I would like to discuss now is around the manufacturing template itself and the key questions that we need to be addressed when making strategic decisions to build your manufacturing strategy. We know that it's complex to design those processes, and your decisions will be impacted by the specificities of your cell therapies, as I already mentioned, but also potentially by the design of an existing facility or the existing capabilities of your CDMO partners. So the key questions are which methods I will be used to perform each step of my manufacturing template. Should I go for open, closed, or an hybrid solution? Where I will be able to perform those processing steps? In which grain environment? Do I need an isolator? How will we ensure the transfer of the cellular materials between unit ops and between all the steps under aseptic conditions? As you know, there is no opportunity for sterile filtration at the end of such a process to remove microbial contaminants. So everything has to be done under aseptic conditions. All of those aspects are definitely interlinked, and your answers and the answers to one question will definitely impact the way you will be able to answer the following questions. So again, I think there is absolutely an opportunity to look at simplifying the process early on as much as you can, because that will give you much more flexibility also to address the specific questions, and will give you more opportunities to be well prepared for transitioning from small scale to commercial scales and from R&D to GMP environment. The manufacturing is one thing. The supply of critical raw materials is another one that you will have to take into account, especially if I take the example of the CAR-T process. You know, critical raw materials or specialty reagents, specialty media, some consumables associated with the technology platform in your template, but it has something to do with some elements like the plasmids used for the manufacturing of your viral vectors and the viral vector itself used to engineer the T cells. So I know that the technology providers in that space, including Merck Life Science, we are all putting a lot of efforts to come up with very good solutions to address those specific needs. I listed on that slide, and I put on the slide on the left some examples of single-use operations or technology platforms that can be used to simplify the purification and formulation of plasmids. And on the right-hand side, I put the example of the ongoing development programs to come up with suspension upstream platforms for the production of viral vectors to ensure high titer, to reduce the manufacturing cost, and to improve the manufacturing efficiency for the production of those viral vectors. And I will come back to that later in the presentation. So all of that being said, let me now move to the last part of my presentation, which will be focusing on the development of fit-to-purpose or advanced manufacturing solutions to address some of these challenges that we just discussed. In other words, how can Merck Life Science help you in developing and implementing your manufacturing strategy? The future of cell therapy manufacturing will bring more templated processes. That's for sure, and which is great, because we need new solutions and new opportunities to manufacture those products in a way that we are not currently using. And we are all looking at more automated solutions, closed solutions. We want to reduce operator risk and risk of contamination. We want to deal with technology which are gentle to the cells, to protect the functionality of the precious cellular materials. We want to remove the presence of residuals that would come from the manufacturing process. We all want ease of use. to come up with simple operations, with intuitive interfaces, with minimal setup and minimal training programs for our operators. We want something really flexible, not only in manufacturing, and definitely maybe less in manufacturing, but more in a PD environment to tune our process in a simple way and to come up with consistent results, more repeatable performance across patient materials, and especially in an autologous setting, having some solutions that could potentially buffer the variability associated with the starting materials. And at the end, on the right-hand side of that slide, if we are very successful in addressing all of these points, there will be a great opportunity to come up with cost savings and cost reduction. And it's really expensive to manufacture cell therapy products, and we have to find solutions to make them affordable to as many patients as we can across the globe. To achieve all of this, I strongly believe that process innovation is still required. And as an example of process innovation, I just wanted to introduce Phoresis as an innovative solutions to process cellular materials in a fully closed and automated manner. You have on that slide 2 examples of those technology platforms. EcoSelect on one hand, which will be used for a T-cell selection process, one step directly down from a leukopak material to come up with purified T-cell population ready to go for the next steps. And on the right side, the ecosystem, which is technology, cell type agnostic technology platform to process concentration, wash, buffer, media exchanges, upstream, downstream in your manufacturing domain. So what it is about, Phoresis, that's the use of ultrasonic waves to capture particles like cells, either by acting on interface between clarified fluid and fluid, mixture of fluid and particles, or by capturing and clustering particles within an acoustic field. It's very gentle because the way we capture the cells within an acoustic field is by just maintaining cells in suspension in the fluid. So those two platforms are still being developed by Merck Life Science. The ecosystem on the right-hand side is commercially available, at least for process development activities. If you want to get familiar with the technology, feel free to contact us. We have demo units available to run preliminary experiments on site with your PD scientist. For the EcoSelect, I will ask you to be a bit patient. We'll bring the technology to the market next year. Another example, if you're looking for single-use manufacturing solutions or high-performing solutions for the production of viral vectors or for the production of plasmids, we have a huge portfolio, including a lot of single-use manufacturing solutions. I invite you to go to our website and to navigate through. You will find a lot of application notes and webinars that can guide you through our capabilities to support these development programs. Let me spend just 1 minute on the Virus Express platform. Our organization put a lot of efforts to speed up the development and to come up with interesting solutions to faster speed to the clinic via reduced process development time and scalability for long-term commercial needs, and better preparedness for regulatory requirements, which can be all realized with the use of this Virus Express lentiviral production platform. So what it is about is a combination of a Virus Express 293 T-cell clones adapted in suspension, used as a producing cell lines, which has been fully characterized and banked under GMP conditions, ready to quickly move to commercial applications, together with a chemically defined media, which is named XL chemically defined Ec293 viral vector medium, which has been optimized for PI, polyethylenimide transfection processes, and for both the expansion of the biomass in a bioreactor followed by a vector production without doing any media exchanges. And this is baked with our expertise around bioreactor processes and our CDMO capabilities and expertise around the manufacturing of these viral vectors at large scales. So feel free to challenge us on the technology. We would be happy to support your development around the production of viral vectors with all of those assets and capabilities. And last but not least, again, back to the critical aspects around developing a robust testing strategy. I would like to emphasize our BioAlliance testing services for cell and gene therapies to help you build your product knowledge and your biosafety testing strategy. So Merck BioAlliance Testing Services is a leading provider of contract services in the biopharmaceutical industry, supporting every phase of the testing, development, and manufacturing process, and offering a comprehensive and integrated package of services all across the modalities in the pharma industry. But we do develop specialized testing service for viral and cell therapy products, just to name a few of them that are very relevant in that industry. Rapid sterility testing, really relevant. The use of replication competent viral particle testing, also very relevant from a safety point of view for all the ex vivo gene therapies using viral vectors to engineer the cells. And another example is the integration site analysis by next-gen sequencing capabilities, again, focusing on the genetically modified T-cell therapies. Also, we have experts that can navigate you through the regulatory landscape across all geographies. They would be delighted to discuss with you those constraints and regulatory requirements to help you, again, really anticipate the need for developing your testing strategy. And I will conclude the presentation today. Thanks for listening up to now, and with 3 key learnings as we went through. The first one is definitely we all understand the complexity of cell therapies, which makes manufacturing quite of a challenge. But to tackle those challenges, I would recommend a couple of things. First, focusing on product characterization, developing reliable QC methods upfront, also securing the supply of key raw materials. All of those aspects are crucial when you start developing your manufacturing template. And on top of it, again, I repeat myself, try to make the process as simple as possible or as complex as necessary, and look at advanced technology platforms which are currently under development, because altogether, I strongly believe this is a strong foundation to streamline the industrialization of cell therapy products moving forward. With all of that being said, I thank you all for listening, and I think we'll be transitioning now to a live Q&A session where we'll be happy to take a couple of your questions. Many thanks.